Okay. So our next speaker is Dr. Wendy Strangman. Dr. Strangman received her PhD from the Scripps Institution of Oceanography, and her postdoctoral research was performed at the University of British Columbia. Now an assistant professor at UNC Wilmington, her research interests center on the application of mass spectrometry-based analyses for the discovery and detection of toxins and other bioactive transformation and permeability, other bioactive compounds produced by harmful algal bloom. Additional research interests include mass spectrometry-based pharmacokinetic assays to assess digestive transformation and permeability of medicinal plants extracts, the discovery of new bioactive natural products produced by marine bacteria from the microbiomes of marine parasites, pathogen-resistant coral microbiomes and deep Arctic sediments, and chemical ecology of Caribbean coral reefs. Welcome, Dr. Strangman, and would you please begin your presentation?
Sure, thanks, Ben. And let me throw this out here. Does that look right? Thumbs up. Okay.
Yep, looks good.
Fantastic. All right, well, thank you, everybody, and thanks to Cambridge Isotopes for the opportunity to tell you a little bit about some of the stuff we've been working on in our group over the last few years. So today I'm going to talk to you a bit. We've got a 1-2 punch on microcystin toxins coming up in this webinar series. And so I'm going to give you a bit of introduction to the world of harmful algal blooms. We're going to switch a little bit to these natural toxins that are being produced. And then I also want to talk a bit about a focus of our group on other molecules that these harmful algal blooms make that either we don't know anything about yet or that might have therapeutic potential. So just as a background to set the stage, harmful algal blooms, I think most of us are aware of what these are at this point, especially those of us that are in the toxin monitoring world. But they're basically what happens when little single-celled algae start growing out of control, and they make these giant blooms that they sit either on the surface of the water or sometimes down in on the bottom of lakes and reservoirs. Those are a little harder to see. They're a little less dramatic, but as you'll see in a minute, they make some pretty gnarly toxins. So some of these can be seen all the way from space, like you can see up here in the upper left. They make these big, dramatic, colorful blooms from their photosynthetic pigments. And they create a tremendous amount of damage and concern to both humans, animals, and the environment in general, which is why we're interested in them, which is why we do research on them, to see really what's going on and what can we do about it. So why do we care? You know, why am I here talking about this? Why does my lab work on this? So this is sort of from the national perspective. Up here in the middle in this map of the US, we can see all of these states that have had this is from the CDC. So in 2021, these are the states that reported having harmful algal bloom toxic events. This doesn't mean that this is where the only place that they are. This is just where they were reported. So reporting is voluntary, testing is voluntary, but pretty much any state that starts testing and starts reporting is going to be reporting these harmful algal events because they're widespread across the country. And they're increasing in severity, or at least as we're seeing in these reportings. They cause human illnesses, they cause deaths to pets and livestock. Over here on the left are sort of the major events recorded by the EPA over the last few years. So they happen across the country in Oregon, Lake Erie. These are sort of the big ones that you can see from space or that have major impacts across states. So for example, you can see down here, Lake Okeechobee from Florida is not represented on the CDC one, even though there were massive amounts of toxins detected, and that's just due to reporting or lack thereof. So annually, if dead pets and sick humans isn't enough to make you concerned about these things, the economic impact of these blooms due to water treatment, due to interruption in tourism, due to property values, if you have a lake house near a harmful algal bloom site, your property is going to go down, can be in the tens to hundreds of millions every year, just from a single HAB event like we have of these major ones over here on the left. So it's a big deal. It's something that we need to think about. It's something that can have some pretty significant interactions, not just on the national scale, but regionally as well. So here's just a few snapshots from various states across the country. So California, Paris Lake, which is in Southern California, is now seeing seasonally that they have these major algal blooms that are producing a variety of toxins. And so they're having to close tourism, et cetera. We see it over here in New York. You know, residents are mobilizing to figure out what they're going to do about their blooms. Illinois, toxins again. North Carolina, where I live, this actually came from Wilmington, the city that my university is in. We had these dogs that were swimming in a local little retention pond, and they ended up dying. These were like really famous dogs in Wilmington. They had their own Instagram pages and everything. So that kind of rocked our little community here. But really, pretty much any state, any city that you guys live in, if you have bodies of water, you're probably seeing these same sorts of headlines, especially in the warmer months when these algae start taking over. So this is sort of why we study these harmful algal bloom toxins, why people care, why we're doing all of this monitoring for them. So the types of toxins that these produce, and this is sort of where we get into the chemistry, where I get excited, where our group gets excited. They kind of generally class into two groups of toxins. So we've got the neurotoxins, and the two most dominant ones of these are saxitoxin over here on the left and anatoxin over here on the right. Saxitoxin is pretty crazy because it's made by both freshwater and marine algae that are very distantly related to each other. But they both make this same crazy structure. It's an extremely potent neurotoxin. So it's actually listed on the Schedule 1 of the Chemical Weapons Convention. So it's a really, really gnarly neurotoxin. The sort of silver lining to this one is if you ingest it and you can get to something like an iron lung, your body will eventually clear it and you can recover. This molecule over here, anatoxin A, which was determined to be the one that killed those poor little puppies in Wilmington, it has very similar symptoms. It's not quite as acutely toxic, but unlike saxitoxin, it's irreversible. So once you ingest it, you're basically a goner. There is no treatment for it. So this anatoxin has become increasingly linked to a lot of pet and livestock deaths from drinking water in contaminated areas. It's also made by species that live below the surface, so you don't necessarily see that green scum on the top. You have to dig down beneath to actually see the cyanobacteria that are making these compounds. So it's a pretty scary one that we're working with. The hepatotoxins, these are sort of the really famous ones. So microcystins are the ones that we're going to talk about for sort of the remainder of the toxin part of my presentation today. They are characterized by this really diagnostic Adda side chain. You don't see that in many other types of cyanobacterial toxins, although you see them in the closely related nodularins over here on the right. And then microcystins are characterized by this two-letter code. So you can see MCLR. That stands for the leucine and arginine circled here in the orange and the blue. Nodularins also have these arginines, but they're a slightly shorter sort of ring system. They've got smaller masses. And then down here, also in this liver toxin family, is the molecule called cylindrospermopsin. And it's got a whole bunch of nitrogens and some charges. It's a lot more water soluble. And it's got this sort of diagnostic hydroxymethyluracil. But it's been linked to animal deaths. It's been shown to bioaccumulate in crop tissues if they're sprayed with water that have been contaminated with harmful algal blooms. So all of these toxins are made by different species of harmful algae, and they crop up all over the country and really all over the world. We see all sorts of different varieties of these. So one thing I wanted to spend a little bit of time talking to you about today is sort of the structural variability in these microcystins. So not everybody who thinks about microcystin toxin monitoring thinks about microcystin LR and sort of three other ones. So the EPA in the US has guidelines for microcystin LR, LA, YR, and RR. So these four microcystins. But what we now know is that there's over 370 different microcystin variants that have been published to date. So there's a tremendous amount of structural variability that you can get within this scaffold. Importantly, as I showed on this previous slide, this Adda side chain over here has a diagnostic fragment. If you're studying it by mass spec, which is one of the most sensitive methods that we have, of 135. Over here, we can see that many of these isomers or these different analogs have modifications here that are going to mess up that diagnostic screening. We also see a tremendous amount of variability in pretty much any part of the amino acid or ring chain here. And this is important because if you have something like an ELISA kit, something that's a non-mass spec method, a lot of these are going to light up the same. But what you can see over here on the right is that these structural changes actually have big implications for how they're going to act in our bodies and in the bodies of animals. So over here, you can see it's kind of a small screen depending on how big your laptop is. But basically, the different bar colors mean different microcystins. And you can see some of them have no activity, some of them have a tremendous amount of activity. And so this difference in activity is just based on small structural changes. So it's very important to sort of understand what you're dealing with. As I mentioned, a lot of the ways that we in our group and many others talk about these things is through mass spec. And so that 135 fragment, which comes from this Adda side chain over here, is really what we use to do our first screening to see if our bacterial strain, our cyanobacterial blooms make microcystins. And so down here on the right, you can see sort of the crude extract or one of the more complex extracts that are made by these cyanobacteria. So each peak is a different cyanometabolite. We don't necessarily, when we do a first extraction, know what any of these things are. So we can start doing the daughter ion extraction at 135, which you see up here on the top. And so anything that has a mass range sort of in the right area for microcystins or nodularians and has a fragment ion of 135 corresponding to that Adda chain, we start saying, okay, this is probably a microcystin, and we start to get concerned. So you can see here from this species that we grow in our lab, we've identified the major compounds as MCLR and MCLA, and then all these other little peaks that have a red star are other compounds that are microcystin variants that we want to know more about. We want to know what they're doing. One of the ways that we look at this and that we've been working on for several years now is the development of fully nitrogen 15 labeled standards. So we have a lot of these cyanobacterial strains growing in our lab, and we've fed them basically as high as we can get purity of N15 labeled media. So we get full incorporation not only into the microcystin toxins, but into every metabolite and every protein and everything in the bodies of these microcystins. So for now, what we're interested in is in the microcystins, and I'm not going to talk much more about this now because the presenter who's coming next, Professor Javier Ortiz, is going to go into great detail on this. But just kind of showing where these standards that he's going to be talking about come from. They come from these cyanobacteria that we're growing. And what you can see is by incorporating N15, this heavier isotope, instead of N14, you get a mass shift, and this can be super helpful, as you'll see next. Where my lab tends to focus, for a long time, the microcystins were just sort of a waste product that we just stored in vials in our fridge, because we're more interested in all of these other molecules that microcystins make. So they make a tremendous number of different chemical scaffolds. And just like the microcystins, each different molecule here is going to have a different type of biological activity. Most of them, we don't know anything about what their biological targets are. So we start by isolating them, figuring out what their chemical structures are. And the next step is to figure out what these are. Are they toxins? Are they things that are going to help us? Could they help cure heart disease? Or are they something that maybe we need to look at for potential birth defects, potential endocrine disruptors, these sorts of things? And just over here on the right is a graph that has been published recently showing how the discovery of not just microcystins, which are in this green line, but all of these different things are dramatically increasing year to year. We're seeing more and more of these different molecules. And this is exciting because they have a lot of really interesting biological activities. So all of these, not just toxins, but potential therapeutic avenues of activity. So that's where we're interested in. Can we find some of these treasures from what are known animals that are just, or organisms that are just known for their toxicity? I was in a meeting yesterday where we're talking about some of these other compounds, since we have them in our lab, both native and fully labeled. We're going to plan on this summer using a combination of quantitative NMR and mass spec and UV, thinking about developing some of these as standards for people to start testing as they're doing their microcystin analyses. And so just like we saw with the microcystins, this is a bloom, a small neighborhood bloom from Durham in 2018. We identified the microcystin, the dominant one, which is microcystin LA, but there's all of these other molecules that don't have that 135 fragment. And so we want to know, some of these are sort of the major compounds. So we're interested in what they are and what they could be doing both in the environment and to humans and animals that are interacting with these blooms. One of the things we do, we're wrapping up here, we take a lot of these blooms, we can analyze them because we're looking for things that are different, that are interesting. And so we use untargeted metabolomics to sort of analyze both bacterial strains up here on the left and bacterial blooms from across the country on the right. And we're looking for outliers. We're looking for things that are different from the herd. That means that maybe they're making a new toxin. Maybe they're a super producer of something and we need to pay attention to it. But we can sort of use this omics level data to help us focus in, especially if we have standards that can kind of ground our analyses to figure out in a big scale, if we're doing large scale sampling, where do we focus our efforts first? And so with that, I hope that I've given you a little snapshot and a little preface to Xavi's talk next about how important harmful algal blooms are, not just for the microcystins that they produce. And I definitely want to drive home the fact that there's more than just these 4 microcystins. There's hundreds of them that we need to be thinking about. And all of these different molecules have different levels of toxicity, potential therapeutic impacts. And the more sensitive our screening, the more nuanced our screening, the more information and the more ability we'll be able to help people in communities that are affected like these blooms. Which you see, this one is just down the street from my lab. So here's a harmful algal bloom with the sign, beware the alligators, but maybe it's actually the algae that we need to be focusing on. So with that, this is my group. Everyone over here in in green has been part of our cyanobacterial team over the last several years. And if we have time, I would be happy to answer any questions.
Thanks very much, Wendy. That was a great presentation. So clearly, these things are much more dangerous than alligators. So I think we can take that away from this presentation. It's actually, no, the work you're doing, as somebody who has visited your labs and seen all the work that you guys are doing, it's just incredible how much we need to know about these things. So I really appreciate your time. Also, sorry I didn't get to introduce the transition from PFAS to microcystins and other cyanobacteria or cyanotoxins. We did get one quick question. What's the enzymatic system in cyanometabolism that governs the diversity of these toxins?
That's a great question. So there's actually 2 different primary mechanisms that make these, especially the peptide ones. There's a bunch of other sort of niche compounds that they make, but the peptides, which are made of amino acids, some of them are made in the ribosomes, like we make our normal proteins and peptides. So those are the RIPS pathway, RIPPS, which is ribosomally produced peptide synthases. And then they also, like a lot of bacteria, normal bacteria, have a totally independent biosynthetic gene sequence called NRPs, or non-ribosomal peptide synthases. So they make them in 2 different ways, and they basically have these modules where they grab amino acids and stick them together. And then they have other enzymes that come in and add other pieces after they make the enzyme chain. They might even add in halogens. They can add in sulfurs and make thiazoles and do all sorts of crazy post-translational modifications. They can stick methyls on the ends of the amines in these enzymes. So there's some very dedicated, very nuanced biosynthetic machinery. And if you're somebody that likes genomes and thinking about biosynthesis, there's a program called Anti-Smash that actually works like phylogenetic trees of biosynthetic gene clusters. So you can go in and look for these enzymatic pathways if you have a gene sequence and see if this machinery is in your cyanobacteria.
Excellent. You definitely know your stuff here. So thank you very much.
Absolutely. Thank you.
We've certainly chosen a good person to do these presentations, or people, I should say. Thanks a lot, Wendy. Okay.